Chairperson, Professor Dr. Yami Bhushan Tripathi sir, and our co-chairperson, Dr. Brijranjan Mishra sir. I request our chair and co-chairperson to kindly proceed toward dais. Professor Dr. Yamini Bhushan Tripathi sir is a distinguished scientist and academic with an illustrious career in Ayurvedic research, medical chemistry, and biochemical studies. Sir has served as an ex-dean of Faculty of Ayurveda at Banaras Hindu University, Varanasi. Sir's pioneering work in Ayurveda and its integration with modern science has significantly contributed to the field of metabolic disorder management, phytopharmacology, and oxidative stress research. Sir, we are deeply honored by your presence and guidance in today's session. Joining Sir in leading this session, we are privileged to have Dr. Brijranjan Mishra Sir as our co-chairperson. Sir is distinguished scientist in expertise in vector-borne disease surveillance and control. He has dedicated his career to improving public health through research and innovation. Currently serving as Scientist C at ICMR Regional Medical Research Center, Gorakhpur, under the Indian Council of Medical Research. Dr. Mishra's work focuses on advancing methods for disease control and elimination. Trained at the National Institute of Virology, Pune, we are honored to have an accomplished researcher with us today. We welcome you, sir.
This session promises to be an engaging one with eminent speakers shedding light on cutting-edge research. We have a fantastic lineup of speakers presenting their research. Now, to begin with the lineup presentation, it is my honor to introduce Professor Dr. Radha Chaube, a renowned professor in Department of Geology at Banaras Hindu University, Varanasi. Ma'am will be delivering a lecture on the topic Toxicogenomic profiling of hypothalamo-pituitary gonadal axis genes in catfish heteropenoids fossils under the exposure of pentachlorophenol. Let's warmly welcome Professor Dr. Radha Chaube.
Can go backward, and this is for pointing.
So, Dr. Chaube,
thank you for nice introduction and respected chairperson and co-chairperson. So, with your permission, now I am going to start my talk on the topic entitled Toxicogenomic Profiling of Hypothalamo-Pituitary Gonadal Axis Genes in the Catfish Heteropneustis fossilis under exposure to pentachlorophenol. So for all of you here, this talk will be little bit different from all the talks what we have listened so far or what all deliberations were there for last 1.5 days. So here we have to focus mainly on the two aspects, that is hypothalamo-pituitary gonadal axis. This is a universal axis which is present throughout the vertebrate system. And the second one is the pentachlorophenol. This is a compound which acts as an endocrine disrupting chemical. So any endocrine disrupting chemical which is present or associated with any living organism, that will lead to the disruption of the whole body homeostasis. So here we are considering teleos or the model system, because we cannot do any direct study on the human beings or a higher group of vertebrates. So many lower group of vertebrates are considered to be a research model for that purpose. So now here, this is a very good example that how teleost or fish are the ultimate source of protein, nourishment. And whenever any pollutant or any outside components or organic chemicals or inorganic chemicals are accumulated in the organism, and ultimate that is consumed by the human being, so that may lead to various disorder. And these all are the organ systems starting from brain, heart, swim bladder, liver, reproductive system, gills, jaw, kidney. So all these system under the exposure of various exposure of various endocrine disrupting chemical that may lead to the disbalance of the whole of the system. And ultimately, we, the consumers, either being a big fish, bird, or humans, they are the victim of all these endocrine disrupting chemicals. So we all know that for a proper functioning of any human whole body, there should be a proper relationship between hypothalamus, pituitary, and gonadal axis. There are certain or almost most of the genes or hormones which are secreted by the lower group of vertebrates, they are also secreted by the higher group of vertebrates. Only the difference arises in the mode of the habitat or area where they are surviving, or the place where they are thriving. Like some, according to the habit and habitat of the aquatic organism, terrestrial, the thyroid hormone is similar, whether it is of aquatic organism or it is of terrestrial organism, and disorder will be just related to that particular organism. So here, under the influence of the environmental stimuli and with the support of the photoperiod and temperature, in the hypothalamus, GnRH is secreted, and that hypothalamus, it regulates the secretion of gonadotropins, which are secreted by the pituitary, and in turn that is activated, acted on the target organism organs, that is either ovary or testes, with the release of the various steroids, and it helps in the proper function of the vitellogenesis, final oocyte maturation, and in case of male, maturation and spermation. So if any endocrine disrupting chemical is coming in this, so what they will do, they will basically hamper whole HPG axis process, and it may ultimately lead to the disbalance or disorganization of the reproductive system and the whole health. So here, now coming to the endocrine disrupting chemicals. Endocrine disrupting chemicals are basically, these are the exogenous compounds that interfere with the production, release, transport, and metabolism, binding, or elimination of endogenous hormones. And these can be either synthetic or natural. So we also produce some endocrine disrupting chemicals. So they can be either polyaromatic hydrocarbons, polychlorinated biphenyls, dioxins, metals, plant sterols, or pesticides. So, and fishes, which are the well bioindicators to understand toxicity of these endocrine disrupting chemicals, as they are valuable to endocrine disruption. And we all know that they are the most common victims, because our aquatic ecosystem, our water reservoirs, the ultimate sink, everything, whatever are the waste to us, they are all disposed into the aquatic system, and that directly or indirectly they are having impact on the organism and to our human health also. So here, nowadays, to analyze or to study in detail what these chemicals or harmful chemicals or EDCs are having impact on either human health or organism health, there are various new omics technologies are coming up, and they are giving us bulk of information at each and every point. For example, genomics. If we want to study at the level of changes in genomes, so we go for genomics. If we want to study at the level of gene expression and under the exposure of toxic chemicals, so then that is known as toxicogenomics or transcriptomics. And another, when we want to study at the level of proteins, so that study in omics is known as proteomics. And when we want to study at the level of metabolism or metabolites, then that is considered to be the metabolomics. And these technologies are used to elucidate changes in gene expression profile after exposure to EDCs. So here, we see that this slide only shows the intricate pathway, which shows us the relation, what is the relation between the brain, pituitary, and gonadal axis. brain and here in the blood, whatever secretions are there, hypothalamus, GnRH, pituitary, and this is a aromatase gene enzyme, and in pituitary gonadotropins. So via blood circulation through the gonadal or after the process of the complete steroidogenesis, it is with the in the liver under the presence of the vitellogenin and these which is synthesized at the liver level, and all these there is a complete intricate relation between these, and then they help in the proper homeostasis. So here the model system we have been using for more than 2 decades in my lab is the Heteropneustes fossilis. So this is the one of the most popular cultured species in the aquatic ecosystem because it is rich in iron content and protein content, but it is very high, having very low fat content. So it's generally given to the heart patients also and very popular among fish farmers also. So this fish is having a different reproductive cycle. His whole cycle is divided into these 4 phases, 5 phases: resting, preparatory, pre-spawning, spawning, and post-spawning. And this, as it is a seasonal breeder, so it is very well maintained according to that, and it is spawned. So what all EDCs in our day-to-day life, we are exposed to number of EDCs, large number of EDCs. So known or suspected EDCs, what we are exposed in day-to-day life are pesticides, ingredients in household products and materials, solvents are there, industrial ingredients are there, and various pharmaceuticals and metals are also there. My focus will be mainly on the pesticides. So there are various classes of pesticides which are being used in agriculture and various carbamate, organochlorine, organophosphorus, and pyrethroids or pyrethrin. So here again our focus will be on the organochlorine, one of the most popular compounds, class of compounds, that is organochlorine. So among these organochlorines, chlorophenols are a class of organic chemicals which are formed by the addition of chlorine. So as the number of chlorine is increased, their potency of toxicity that is also increased. So it is widely used in industries and agriculture, and mainly it is deposited in the environment due to the anthropogenic activity. Annual worldwide production extends to 200,000 tons, and it is banned in most of the countries. But in India, such type of consideration has not been taken. So another is that degradation is very complex of this molecule due to the presence of aromatic ring. Degradation is usually incomplete. pH in water, soil, and sediments. So there are 5 basic types and 19 different chlorophenols. So, and they are also considered to be xenobiotics because of their anthropogenic origins. So, and most potently, they are the endocrine disruptors. So, there are various types of chlorophenols, and among those are the monochlorophenol, dichlorophenol, trichlorophenol, tetra, and pentachlorophenol. So, here my focus will be mainly on the pentachlorophenol. So here you can see that as the chlorine molecule is being increased in the benzene ring, its potency of endocrine disruption goes on increasing. So now pentachlorophenol, it is a stable, non-corrosive synthetic compound which is used as a fungicide, biocide, pesticide, and it is generally used in disinfectant. It was first produced in 1930s and marketed under various nomenclature and names, and widely employed in synthesis of dyes and wood preservatives, disinfectant, insecticide ingredients. Due to its extensive use and ubiquitous appearance in the environment, PCP is of most important concern, and its stable aromatic ring system and high chlorine content makes PCP not prone to degradation, and it is considered to be the priority pollutant, and its generalized use has led water contamination, and its indiscriminate use has resulted in considerable human exposure to PCP. So here, this slide is just to show what are the different sources of the PCPs, which I have already mentioned. So here you can see that it is used for in wood preservative, as a wood preservative, herbicides, disinfectants, then dermatological medicinal treatments, biocides, insecticides, etc. So PCP and its derivatives, when they are associated with the aquatic environment, toxicity of PCP depends on the environmental factors such as its how much concentration is available, what is its pH value, absorption to suspended solids, temperature, biodegradation rate, and photodecomposition rate. So here, according to our study and earlier preliminary studies, we have shown that PCP is not considered as a persistent contaminant at low concentration due to the photochemical degradation and microbial breakdown. But it is hydrolytically stable at pH between 4 to 9 because it prevents hydrolysis, a major degradation process. As the pH and salinity decrease, the compound dissociates and rapidly photodegrades in surface water. So these are the various sources, and there are certain natural source of EDCs and synthetic source of EDCs, and ultimately they are associated with our ecosystem. And these are the various sources like hospital, human activities, agricultural pesticides, from where we all are depositing all these into the aquatic ecosystem. So, for this, our study of toxicogenomic profiling, our objective was to study key important genes which are associated with the hypothalamus pituitary gonadal axis. So, for that, we collected male and female catfish, Heteropneustis fossilis, during a specific breeding phase, that is pre-spawning phase. That time vitellogenesis is very high, and it is under breeding phase. So, and male and female were divided into various groups, and then they were treated with the various concentration of PCP. After completion of the experimental, tissues like brain, pituitary, and gonad were taken, and serum was also there. But in the here, I will only present the data related to brain, pituitary, and gonad, and then qPCR analysis for gene expression was conducted with specific primers of this particular species only, because we have already cloned and characterized 27 genes which has been present in this particular fish model system, and we can directly go for the qPCR analysis. So here we can see that in case of brain, in both and female, male and female, under 30 and 60 days of exposure, there is a concentration dependent increase or upregulation in the GnRH2 expression. However, in case during 60 days, we can see that in case of male, there is no significant variation. And in case of brain aromatase, generally in steroidogenesis, it occurs in gonad. All of you know that it occurs in gonad, but steroidogenesis is a process which occurs in brain also. So brain has got whole body brain two forms. In human, mainly in case of human, only single type of aromatase gene is there, but in teleos, two forms of aromatase is present. One which is exclusively expressed in the brain, that is known as brain type of aromatase, and one that is exclusively present in gonad, that is known as gonad type of aromatase, and that is expressed by CYP19A1B and CYP19A1A. So here we can see that brain type of aromatase is also upregulated in both male and female in a concentration and duration dependent manner. However, when we go for 60 days exposure, this aromatase, brain type of aromatase, is decreased in both male and female. So now coming to the pituitary. So for gonadotropin, we showed that LH beta and FSH beta both are at 30 day and at 30 days, they are upregulated in both male and female in a concentration dependent manner, whereas LH beta was decreased in both male and female in a concentration dependent manner at 60 days of treatment. Coming to the ovary part, so in ovary, estrogen receptor alpha, beta. So estrogen alpha show differential expression. In case of male, it was upregulated, and in case of female it was downregulated, and at 30 days, and during 60 days of exposure it was again downregulated in case of female, and differential expression was, dose dependent expression was observed in case of male. And estrogen receptor beta, that was also upregulated in case of both male and female, and estrogen receptor beta in case of after 60 days of exposure, that was also shown a differential expression with respect to the concentration. Vitellogenin gene expression was upregulated in both male and female in after 30 days, and it was decreased after 60 days of exposure in case of female, and it was upregulated in case of male. So in this way we can conclude that in case of male, where vitellogenin expression should not be there basically, generally, but due to the exposure of these coming EDCs in male, it has been upregulated. Now coming to the other, like ovarian type of aromatase, means ovarian steroidogenesis, that is also showed differential expression in both male and female during 30 and 60 days of exposure. Similarly, we have seen that 3-beta-HSD expression level was also upregulated in case of both male and female after 30 days and 60 days of the exposure. So in a nutshell, we can summarize that PCP is a potent endocrine disruptor capable of interfering with hormonal feedback regulation via HPG axis. The use of these toxicogenomics tools is vital to access pollutant responses in aquatic organisms. Overall, in the present study, we have shown that there was significant alteration in the expression of GnRH2 CYP19A1B in brain, and LHFSH beta in pituitary, and estrogen alpha beta vitellogenin CYP9 ovarian aromatase, and 3-beta HSD genes in ovary. Expression pattern, what was observed mainly was as a dose dependent and duration dependent changes compared to the control, and there was significant sexual dimorphism. Means all the study we also showed that there is a sexual dimorphism expression pattern was observed, indicating that both male and female species are affected widely. So thank you. Now I would like to thank you. And if there are any questions.
So much, ma'am.
Thank you, ma'am, for your valuable insights. So if there is any question, 1 or 2 questions we can take. No questions? No question. So I have one question. Anybody have?
Ma'am, that was wonderful presentation. My doubt was in your second last slide, you were discussing about the results of ovary. So in that also we have male and female. So is that what exactly would be the reason? Whether it is a organism,
actually,
the fish is
that ovary, no, in place of that ovary, I should have written gonad. So that change I could
yes,
not do.
okay,
Yeah,
okay.
gonad.
So
That is I just
okay, okay.
Because it is a gonad specific genes. Whatever they are
Okay,
gonad
okay,
specific.
ma'am.
So thank you. So I have one curiosity, just to know how your findings can be utilized for environmental health purpose.
Yes, sir. Good question, sir. It is because whatever all the genes, so under influence of the environment also, we are also studying, because as we know that all these environmental contaminants are there. So one thing is that we can study such type of work can be studied at the level of the pollutants also, and at the level of climate change also, because already we have seen that teleos model system reproduction is under the control of the photoperiod temperature. So if we go for certain, likewise heavy climatic changes are there nowadays. What we expect, climate is showing us. So when we go for altered photoperiod and temperature under these circumstances, along with the pollutants, so that will also give us a different sight on these parameters. So that can be very widely used.
Thank you.
Thank you so much, ma'am. I now welcome our next speaker, Dr. Shinos Skariachin. Dr. Shinos Skariachin is a faculty at the Department of Microbiology at St. Peter's X College, Rajapuram, Kerala. So work focuses on application of computational methods in drug discovery, particularly in fight against antimicrobial resistance. Today, Sir will be presenting his views on the topic computational drug discovery for combating antimicrobial resistance and escape pathogens. Please welcome Dr. Shinos Skariachin.
A very good afternoon to one and all present here. Respected chair, respected co-chair, and the esteemed dignitaries, scientists, research scholars present in this esteemed auditorium. First of all, I would like to thank Professor Dr. Edathil Vijayan sir and the organizers, especially Professor Sunil and Professor Amit, for giving me this wonderful opportunity of presenting some of the work that is in this esteemed SBTI conference. So I am glad to see that most of the audience are the students. So I would like to tell you some of the contributions of the students. After this talk, you should be getting an insight how students can do in the research. So that is what I am going to show you. And you know that we are going to address on one of the most important issue, that is the antimicrobial resistance. So what is the role? A couple of speakers, during this time of this talk, couple of speakers have, they have clearly mentioned the role of bioinformatics and computational biology, especially computational drug discovery to cope up with the various, the life-threatening infections, and what is their role they have identified. So I would like to talk on this particular talk, the computational drug discovery of combating antimicrobial resistance in the one of the important, pathogens called the escape pathogens that definitely you will be knowing. So you know that antimicrobial resistance is one of the most potential problem. According to WHO, you know that every 3 minutes one child is dying, and we are having several type of issues, and we are having approximately 7.5% of decline in the livestock by 2050, and this is going to be one of the potential problem nowadays we are going to witness. I think you remember the last G20 nations, the summit has happened in the Delhi under the leadership of our Honorable Prime Minister. The India has debated one of the most important aspect of antimicrobial resistance. In that summit report, if you look, they have developed, the nations, they have developed some of the guidelines of combating this antimicrobial resistance actually. So, when you are talking about the antimicrobial resistance, now WHO, they have given the list of priority pathogens. So 2017, the first list was announced. Then, that 2024, the list has, they have revised. Then there are critical group, high group, as well as the medium group that is there. Especially when you are looking the critical, the priority number 1 pathogen like Acinetobacter baumannii and Pseudomonas aeruginosa, such a kind of critical pathogen, they resist the last resort of antibiotic like the colistin as well as the carbapenem. Then you will be having high group as well as the medium group you will be having. So now we know that when you are talking about this one, the attraction is the escape pathogen. What is escape pathogen? For the students, I would like to mention this is a short form of that is Enterococcus faecium, Staphylococcus aureus, then Klebsiella pneumoniae, and Acinetobacter baumannii, Pseudomonas aeruginosa, and Enterobacter aeruginosa. The collective that short form called escape pathogens, and this pathogen contribute maximum mortality rate in among antimicrobial resistance. So this is the global status of antimicrobial resistance. you know that I told you 2015, it is going to be, the death will be overcoming than the that compared to cancer and the cardiovascular infection. So lot of problems are going to happen, in which Asia is going to be the #1 continent in the world. A lot of mortality per 10,000 population is going to happen. This is the one of the latest statistics of WHO. Then for the students' sake, students should know what is the importance of and what is the causes of this antimicrobial resistance. Although you know that the causes of antimicrobial resistance, the main problem is over prescribing the antibiotic, and the patients are not completing finishing their treatment within time. Then the overuse of antibiotics and the lack of hygienic and the poor infection control and such kind of thing. All those things that is very well narrated by the WHO. And how antibiotic resistance is spread across the globe. You know that it is mainly spreading through the drug-resistant bacteria in the gut and through food, and as well as the drug-resistant bacteria spread to the patients to patient due to the poor hygiene. And the patients are spent in the hospitals and clinic, and the antibiotics which are given to the clinic which resist the drug-resistant bacteria developing the gut, and similarly antimicrobial resistant genes are spreading. Now do you know that what is the general mechanism of antibiotic? This is, undergraduate students, the BSc students, they'll be knowing what is the mode of action. There will be most of the antibiotic, the mode of action will be, there'll be cell wall synthesis inhibitor, some antibiotic. Then one of the most important thing in the bacterial system is folic acid metabolism, the inhibition of folic acid metabolism. Then cytoplasmic membrane structure inhibition. Then the enzymes like DNA gyrase and RNA long-acting and DNA-directed RNA polymerases, mainly the protein synthesis machinery that is inhibited by the several antibiotics. So these are the mechanism. I have given this a lot of examples of antimicrobial. How, say for example, rifampicin. So rifampicin, so that RNA-directed RNA polymerase inhibitor. Similarly, that nalidixic acid and ciprofloxacin, novobiosine, they are quinones, DNA gyrases inhibitor. So such a kind of active mechanism they are having. And how the microorganisms, they develop the resistance. The mechanisms are very well studied. They will be having the antibiotic modifying enzyme, then the target protein production enzyme, the efflux pump mechanism, the target site modification, the target bypass, and similarly decrease the efflux pump. All those things is very well studied, that people are knowing properly. Then, there are a lot of, don't think like we don't have any strategies. We are having a lot of tremendous strategies to combat antimicrobial resistance. So, the antimicrobial peptides, one of the very marvelous area of, work, antimicrobial peptides, then host-directed therapy. During the COVID-19 time, people have been heard about the word drug repurposing of the FDA-approved drug. Then similarly, you'll be having vaccination. Then you will be having the phagy therapy, very excellent method. Phagy therapy and the anti-virulence drug mechanism, and people are working on CRISPR-Cas9 antimicrobial agent. So these are one of the most important technologies. So my concept, we are talking about what is the role of bioinformatics. Now we know this is the antimicrobial resistant diagnostic in the market. By 2030s, people should spend a lot of their investment to decrease the antimicrobial resistance and infection. They should need a treatment mechanism for the antimicrobial resistance. So that is, now we are in 2025, 4.6 billion US dollar now they are spending. So now that is going to be enormously increased by 2013, because that is the danger of this antimicrobial resistant problem in the healthcare nowadays. Now, when you're talking about drug discovery, so this is again for undergraduate student, drug is a very, very toughest things to discover. It is a very complex, extremely complex process. So if you talk about drug discovery, initially, when you look for a lot of initial molecule you'll be having, then you should undergo the preclinical, then you should be having the clinical level, phase 1, phase 2, and phase 3 clinical level. Then you will be having the evaluation, the approval by the FDA, and finally, that is subject to the approval. So it will take a lot of time. It will take a lot of time, and that if a company, they spend more than €1 billion, so finally, if the company is very lucky, they will be able to come up with 1 or 2 antibiotics. So at the end, that is what is the microorganisms are resistant so fast. So the problem, that when you are talking about this one, this concepts are so complex. Initial phase 1, phase 2, phase 3. We need to do with so much of assays, lot of initial wet lab work. So this is here comes the problem. It will take a lot of years, nearly 15 to 16 years it will take. And here comes the role of bioinformatics. And you know that whatever throughout so many speakers have mentioned about the role of bioinformatics. Initially, the initial part of these particular studies, especially the screening of the targets, the screening of the ligands, screening of the small molecules, screening of the antimicrobials, all such kind of things, we can do bioinformatics analysis, especially for structure-based drug discovery, ligand-based drug discovery, similarly, system-based drug discovery, the target identification, target validation, lead identification, lead optimization, and preclinical and clinical level. Initially, all those things you can come up with the bioinformatics, and you will be able to come up with the ideal molecule with good drug-like features, pharmacokinetic features, and the toxicity criteria, etc. So this is the importance of bioinformatics. Then when you talk about, that the scope of bioinformatics, especially computational biology, bioinformatics 2024, if you look the Nobel Prize for Physics and Chemistry, the Nobel Prize for Physics, the people got a Nobel Prize in Physics, the machine learning. Okay, especially neural network. Neural network, we use a lot of bioinformatics tools as a basic mechanism for the prediction of the secondary structure, etc. Then if you look the Nobel Prize for the chemistry, the very outstanding study they have done by the David Becker and Demis Hassabis and John M. Bember, because they have developed a massive, excellent tool called AlphaFold. You know that AlphaFold, if you look at the paper, Nature, the Science, the top level journals, they have been using AlphaFold server for the initial prediction of this particular molecule. So this is the power of bioinformatics and power of computational biology coupled with artificial intelligence and machine learning, dear students. So this is what I wanted to tell. Now you know that when you're talking about viral infection, and in the we ourselves we have started a small research topic in Frontiers in Microbiology, and I was the editor for that, and we have written what is the role of bioinformatics, computational drug discovery in the emerging viral infection. We have been witnessing past 5 to 6 years, including COVID-19, Nipah, okay, all those things, Marburg virus, all such kind of things we have been witnessing. Lot of viral infection. What is the role? This is available. You can read. Then similarly, another important thing, the editor of Future Virology, they have requested, he sent me an invitation to write an editorial on this particular things actually. So what is the scope and discovery of potential therapeutic agents using bioinformatics as a potential next generation therapeutic? So this is the another important, again it is available. Now what I'm trying to tell is what is the overall workflow of computational biology. So many scientists, they have already discussed here, that you will be having a beautiful target identification. You need to identify drug discovery. What is the thing? You need to identify the target. Target identification is a very, very complex procedure because it requires a crystal structure or NMR structure or so-called the cryo-electron. Yesterday, they have mentioning about cryo-electron microscopic analysis. Okay, so all such kind of excellent target identification method, and we are having databases for screening the structure from the various type of target. Then we are having the databases to scrutinize or analyze the ligand molecule, and the structure is not available. Then what you need to do? You need to you need to make your own model. I told about AlphaFold. Okay, similarly the homology modeling or ab initio modeling or threading, such a kind of approach you can make, and finally you can validate. And finally, people do only with the basic modeling. Basic modeling is not sufficient. The ultimate aim of this bioinformatics is to successfully implement. It should be implemented in the wet lab. I'm not telling like bioinformatics is everything. It should be wet lab. Okay, wet lab is the final and ultimate outcome. So what do you need to do? You should get the accurate outcome for that actually. So definitely we need to do molecular dynamic simulation. Yesterday somebody already mentioned. Okay, so this is the and the question is where you'll be getting the molecule. The previous speaker's chair itself, they have mentioned about the scope of small molecules, especially when you're talking about the plant-based molecule, the natural molecule. This is one of the main focus of this particular conference. That is the reason I am highlighting this one. We are having beautiful databases. Okay, instead of going for mass GCMS data, MS data, all such kind of data, any molecule you do, just you will be getting the entire 3-dimensional structure in the database. There are a lot of databases like PubChem, similarly DrugBank, then ChemSpider, then KEGG, then similarly Sing, then you will be having another natural database called Coconut. Similarly, you will be having Supernatural too. Then similarly, you will be having NPAS, many more databases. Then some of the commercial manufacturers also, they'll be having database. So this one, this database can be used. Then the ultimate flow, this again for the student, the ultimate flow of structure-based drug discovery. So what is the important thing? You need to take a small molecule that is not sufficient. You should make sure that is why where we are using bioinformatics. Okay, initially, you know, when you do the conventional experiment, you need to do lot of wet lab, you need to do lot of studies. It will take lot of time actually. So instead of that one, why can't you use predictions? Okay, why can't you use modeling with good quality software? Okay, so this is the prediction of drug likeness. We can do the drug likeness prediction. You can do it actually. Then similarly, the speakers in the earlier time, they have told the pharmacokinetic features, the absorption, distribution, metabolism, and the excretion, such kind of studies. The prediction of toxicity, all those things are only we are spending maximum time for the wet lab. Why can't we use these particular things by bioinformatics approach? And finally, you do the docking studies. The morning, Ramesh sir also, he has shown some of the studies about the docking result actually. You have seen, okay, how molecular docking is, and he has mentioned, earlier people used to do wet lab. Now, then people have already, now we are doing then the reverse order. After that, we are doing the in silico. Now we'll be doing the in silico first because that is the power of computational biology. Because we can minimize the actual cost, time, complexities, all the infrastructure that is required in the laboratory by using within short span of time. 5 lakh work, 5 years work, you can complete it within 1 or 2 days. That is the power of artificial intelligence and machine learning, as well as the computational biology software and tool. Then finally, you know, people do just a docking. That's not sufficient. You need to, because it should work in the in vivo system, isn't it? It should work in the wet lab. So it should say that, you know, because the docking, we are not taking care of the lot of other surroundings, important molecular dynamics parameters. So there will be a lot of binding mechanism that is happening. So you need to do the molecular docking simulation. MD simulation you need to do. Finally, obviously I told you, bioinformatics is not the end. It should be validated by experimentally. So that is the most important thing. Then I told you for tracking drug likeness or pharmacokinetic features, there are a lot of typical rules, Lipinski rule, Selam rule, read-like rule, MDDR, like lot of filters and molecular, we call it as molecular descriptors. Okay, so that is available. Similarly, we are having for testing, checking the toxicity. We are having beautiful, you know, methods like industrial absorption. All those type of, all this type of theoretical models are available for testing. Then where will be getting? There are open software. There are commercial software. We will be having free open software that with high accuracy software that is recently published 2022-21 nucleic acid research, such a kind of a reputed journal. It is published. Now you know that when I am coming to my work, so this is what the students I wanted to seek your attention. I will show you a couple of you people. Okay, couple of students of your size. That means to say they are not even PG student. In the research which I am showing here, not even a single student is PhDs. They are just undergraduate BSc and MSc student. That is why I wanted to see your attention here. So what wonders you, what you can do it. So here we taken a target called, for example, Acinetobacter baumannii. This is what one of the important organisms, the priority #1 pathogen. Okay, and we have seen there identified a lot of molecular biology and biochemistry is very, very important, basics, and we have identified the homologous recombination pathway. Then we identified that there is an important protein called RECA protein RECA. So this is a DNA strand exchange and recombination protein called RECA. This is the main important thing. This is connected by several important targets. Obviously, you can take this particular thing as a drug target. But the question is, there is no crystal structure. Okay, there is no crystal structure. Then similarly, another important target called pyrene, that is orotate phosphoribosyl transferase. So that is also having a profound mechanism. Because of lack of time, I'm not going to go this one. This is the pyrene this is pyrene. This one. This particular thing is pyrene, and you can see that what is the enormous amount of connection they have made. Unfortunately, both the things they don't have crystal structure. Without a 3-dimensional crystal structure, we cannot do drug discovery. We cannot study the binding studies. So what I have done? I have done the modeling. Okay, so this crystal structure we have modeled. Okay, I don't go for the details. We have modeled both the pyrene and RECA model we have made. then we have validated by a lot of bioinformatics software. So just like that, if you model, that is not sufficient. You need to prove that tomorrow somebody is crystallizing the structure or doing some experimental studies like, your cryo-electron microscopy, accurate structure you will be getting. You should not have a variation, whatever it is coming, that particular structure. Then only your prediction will be making good sense. So what do you need to do? You need to, we have done a lot of validations. I don't go for, because this is the core bioinformatics. Again, a couple of times they have mentioned the Ramchandra plot analysis and such kind of thing. Again, I won't go for much details. Both the model we did it actually. Then the next objective is, I told you Acinetobacter baumannii, standard antibiotic that is resistant. Last line, carbapenem and cholesterol. Doripenem, meropenem, ertapenem, pharopenem. All such kind of antibiotics is the resistant Acinetobacter baumannii. Now the thing is like, how will you identify novel therapeutic substances? So this is the thing, supernatural to one database we identified. We did a virtual screening and we did the entire database molecule we have done. Then what is done is we did the virtual screening analysis. Then we did a molecular docking. Then when we did a molecular docking, I identified that one molecule called epiesterol in that particular database that is showing excellent binding with both the target. Both the target almost the same binding site. Okay. So then what is done? The binding energy, everything we calculated, we studied. Okay. So because of students are sitting here, that is why I am not going for the much about the structural basis over here. Then what you have done? Then we did a comparative studies. So we did how the pharopenem. Okay. So pharopenem is interacting with their target. Okay. The binding with the conventional antibiotic pharopenem. So I could find out that my molecule, the molecule which I screened, showed a better binding and better dynamics and better stability, better weak interaction to the amino acid that is present, that pharopenem and its non-target like penicillin binding protein. So that is the better finding. Then we did the preliminary in silico studies. We wanted to validate. This is the MD simulation we did actually. The MD simulation for initially 100 nanosecond we did it, and we purchased the small molecule. Then we did the wet lab experiment, and you can see a lot of plots. So then what we have done is we could see that the molecule what I have suggested, specifically against the PIRE and PIRE, the RECA protein, that specific mechanism we are going to detect, and initially we could see that will be having some kind of excellent inhibitory activities. So this is what we have seen. Okay, then this work is already published. I told you all this work is done by undergraduate students. Okay, now my student on this particular paper is published in Infection, Genetics and Evolution. Similarly, the concept we have explained in critical reviews in microbiology. So similarly, then another student came, sir, why can't you use another target? I promoted her, I motivated her. Then you take this one. Then she come up with a target called carboxymonodecarboxylase. They're having some substantial, role in this particular pathway. Generally, they are involving in benzoate pathway, and they are having some important pathway. And this is not studied also. There is no crystal structure. Then, as I said in the previously, then we have done a molecular docking. Then we did a molecular modeling, and finally we have done lot of bioinformatics analysis. And we could see that we identified some natural molecule from plant origin, and you can see that these are the excellent binding pocket, docking paths, and we have identified the best molecule. Then two molecules that is said to be, one is the thymoquine and quercetin. They are showing much better binding energy than the normal usual, the carboxymuconolactone of this particular charge. So this is, you can see that the binding pocket, when they are co-crystallizing the structure, they are the same thing, they are binding in a very effective way, and with their usual substrate, these two molecules are binding in a better way. So this is the way we propose the mechanism. Obviously, we have done a lot of MD simulation and post-MD analysis. All such kind of things we have identified. We did it enormous amount of studies. I won't go for much detail. Finally, this has come up with in a good journal, Computers in Biology and Medicine. So this particular student called Shraddha Rana. Okay, she's from Pune, and she's now when she was doing her MSc microbiology, she came to me and she was done this particular work actually. Then again, we wanted to identify some of the potential drug target. The outer membrane proteins are the most predominant potential target. Okay, potential target. So then what we have done is we identified this particular target. Both the target we have identified, and we did a metabolic pathway analysis. We did a docking study. This is with the carbon fullerene conjugated with some of the molecules. So the student came because I wanted to motivate her. Then I have done this particular thing because of lack of time. I won't go for the details. Then we did the MD simulation. Again, so much of MD simulation we did it, and post-MD analysis we did it. Finally, that particular paper also it is when I took JBSD, the Journal of Biomedical Structure and Dynamics, I think again same Acinetobacter baumannii. Then our focus was to understand the antimicrobial resistance mechanism of Pseudomonas aeruginosa. So Pseudomonas aeruginosa, as you know that this is one of the well-studied organisms. Okay, so the main mechanism is a biofilm formation. The biofilm formation is, you'll be having the flagellar assembly, bacterial secretion system, cyclic GMP mediated pathway, RASM pathway, then quorum sensing pathway. We are really interested to studying in the two important things. One is quorum sensing pathway and GSM pathway. Then we have identified two important targets. The one is response regulator, GAPK. Then similarly, transcription activator, RHL. Both the things there is no structure. As I said, we did the molecular modeling studies and we validated and we did the screening. So initially we have screened a molecular library of 10,000 molecules. When we checked the drug likeness, 54 molecules are qualified. Then again we narrowed down 32, the ADMA. Then finally 4 molecules qualified. The toxicity, all those things. That particular small molecule we have taken, and all those things are from plant origin. And we have identified the interaction modeling, and you can see that these are the two, celestial and similarly rotinol. So these two molecules showed the better result. And when we comparative control, we have used as a meropenem. This is a standard antibiotic used against Pseudomonas. So you know that then this is the best binding pocket of all those processes I have told. Okay, in comparison with the control. So now what we done? We did the MD simulation, lot of MD simulation work. Finally, we proved that the molecule is really stable. Now we are in the pipeline of whatever I'm showing. Since the student work, experiments are going on. That is further experiment studies for validation of this one is going on actually. Again, this one is the outcome of the studies. We have done the infection, genetics, and evolution. We have studied. We have published. Then folio microbiological. This is one of the best cited paper. Within 1 year, we go to more than 150 citations actually for this particular paper. This is already available. So these are the another important things. So because a lack of, similarly, another target, again, we have used I won't go for much detail, same kind of approach that also we have communicated and published in the Biomolecular Structure and Dynamics. So finally, I would like to conclude what I wanted to tell the students. So that is why all the initial speakers, from the especially the Dr. D.P. Singh sir, Balram Barwa sir, all our esteemed speakers have told you please listen and ask questions. Especially Ramesh sir was motivating the students. Okay, please ask questions. So this is the way. If you look the conferences and symposium, you can come up with if you do such a kind of an early stage, if you are coming some breaking studies, it will be helpful for your career. So that is what I want to tell you. I would like to conclude the multi-drug Acinetobacter baumannii antimicrobial resistance is a potential problem. Then we have screened several type of lot of molecule by bioinformatics, and similarly epiestrol against racea and pyre, similarly carbon fluorine, and similar transcription activators against and response regulator as a probable drug target for Pseudomonas, celestial, glutinol, all the small molecule, small molecule we could be using, and a natural molecule we can use. And this is what I wanted to conclude. And this is the student which I have told. All the students are this the initial work I have undertaken by when I was in Bangalore, when I was taking the Biotechnology student. She's a Biotechnology student. After this paper, she got a scholarship and she has completed her PhD and postdoc in the University of Graves, France. Similarly, another student, Roshni Ravishankar, I was her BTech mentor, okay, and she published that particular paper and she got into the Hamburg University of Germany. She completed her postdoc now. And now another student, Vaishnavi Badridas, IRS University, Denmark, she has completed. And no, this is the Sneva Basuraj. Another paper author, undergraduate student, she got the scholarship, full funded scholarship in Karolinska Institute of Sweden, and she completed her this particular thing. So all those, and Aditi, she's studying in IIT Madras, and she completed her PhD. And I have her my collaborator, and this is the student I have told, Shraddha. And when I was going in an online talk, she attended and she contacted me through mail. Sir, could you please provide an opportunity? I have never seen her. Okay, and I have made a I have made an online interaction we have made, and we go to she got this one, and she is in ACIR NCL Pune. She's working as a PhD student actually. So finally, I would like to sincerely thank he's some sort of vendor like a person actually. Professor Dr. M. Radhil Vijayan sir, he has given me an opportunity, and similarly, the great support of Dr. Sunil as well as Amit Kumar Dubey. They have given outstanding work along with the excellent pool of research scholars and the team. Okay, and each and everything they are taking care of. I thank their great effort. So this is what I would like to thank. And finally, if you are having such a kind of papers, I would like to welcome all of your submission. There is a journal called Computers in Biology and Medicine. That is a Q1 ranked journal. If you talk about the impact factor, that is 7. And I am the editor-in-chief of this particular journal, actually. I'm handling bioinformatics and computational.
So thank you, Dr. Sinas, for your insights on AMR. So now session is open for questions, please. No question? Thank you. Thank you, sir.
Thank you, sir, for your engaging presentation. Our last speaker for this session is Dr. Gautam Anand. Dr. Gautam Anand, a leading scientist at Ben Gurion University of Nagpur, Israel. Such research focuses on cytokinin response and sensing and fungi, a critical area in understanding plant growth and disease resistance. Sir will share his expertise on the topic cytokinin response and sensing in fungi. Let us give a huge round of applause for Dr. Gautam Anand.
Do you have color mics? Please provide. There are notifications.
Hello, hello. Am I audible? Hello, am I audible? Hello, hello. Hello. Am I audible now? Okay. A very good afternoon to everyone, and I'm thankful to the organizing committee for allowing me to discuss my research work, a part of my research work. And as Radha ma'am said, my talk will be different from other speakers. And today I'll be talking about plant hormone cytokinin response in plant pathogenic fungi. And the pathogen of our study was Botrytis cinerea, a grey mold pathogen. It's one of the most devastating plant pathogen, having more than 1,200 plant hosts. It infects fruits, vegetables. Recently, it has been found as a host on medicinal and aromatic plants as well. It can infect leaves, stems, and it's a very important post-harvest pathogen. In China, recently they found they have found that Botrytis cinerea has been a host to many medicinal plants as well. Plant hormone cytokinin, you must be knowing it's an important plant development hormone responsible for organogenesis, cell division, meristem function, nodulation, and recently our group and others have reported that plant hormone cytokinin could also induce immunity in plant against fungal pathogen, either by inducing the signaling pathway, salicylate or jasmonic acid pathway, or by modulating the content and distribution of pathogen recognition receptors in plant. So just to make sure that induction of plant immunity and the modulation of PRR distribution is the only way plant cytokinin is able to inhibit the fungal pathogen, we did an in vitro test to make sure that there is no direct effect of the hormone on the pathogen. But to our surprise, we found that cytokinin was able to directly inhibit the plant pathogen botrytis in vitro. And not only Botrytis cinerea, it's a necrotrophic plant pathogen, Botrytis cinerea, aerial pathogen infecting leaves, stem. We also checked its effect on Sclerotia roxi, which is an airborne necrotrophic pathogen causing stem rot, Fusarium oxysporum, which is a hemibiotrophic soil pathogen, and cytokinin was able to inhibit the growth and virulence of all these pathogen of different lifestyle and different mode of action. The cytokinin we used in our study was 6-BAAB. It's a synthetic cytokinin because it's stable. But we tested the effect of natural cytokinin as well at different concentrations, like zeatin, kinetin, transzeatin, PDZ, and all were able to significantly reduce growth of Botrytis cinerea pathogen, both mycelial area also and dry weight. This slide shows the physiological level of plant hormone cytokinin, like transzeatin, isopentenyl riboside. And if you roughly measure the physiological concentration, it comes to around 400 nanomolar of transzeatin, but this is just the single cytokinin. There are many cytokinin. The purpose of doing this was to make sure that even the physiological concentration which is present in the leaves is also able to inhibit the fungal pathogen. We checked the effect of cytokinin 6-BAAB on sporulation and spore germination, and we found that sporulation, spore germination, and in fact germ tube length was also significantly reduced by the cytokinin. To gain a mechanistic insight of how this cytokinin is inhibiting the fungal pathogen, we did a RNA-seq analysis of the fungus treated with cytokinin, and the transcriptomic data we found, it showed that there are a lot of cell cycle pathways were downregulated. DNA replication was downregulated. Endocytosis was also downregulated. Also, we found some of the virulence genes were downregulated. The virulence genes in fungal pathogens are not very well annotated. So we made a list of our own virulence genes and we found that a lot of them were downregulated in the transcriptomic data. To confirm our RNA-seq analysis, we did some cell biology assays and tried to measure the cell area, cell length, and number of septa. Also the nucleic acid content, because the cell cycle was found to be downregulated in the transcriptomic data. And we found that all these parameters were significantly downregulated when the fungus was treated with the cytokinin. We have used chalcofluor staining for the septa and the cell membrane, and Hurst for nucleic acid content. Apart from the cell cycle and endocytosis gene, our transcriptomic data suggested that a lot of cytoskeleton genes are downregulated. Also in the qPCR analysis, we found that some of the fungal cytoskeleton genes were downregulated. To confirm this transcriptomic analysis, what we did was we transformed our wild type fungus with life-actin GFP construct. Life-actin or first F-actin is the first cytoskeleton that is formed in the cell, and it's a hallmark of growing hyphae that sorry. Sorry. Back Kamal. Yeah. In the growing hyphae, F-actin is found to be localized at the tip of the hyphae. You can see at the tip, this is GFP life-actin. So what we did, we used our transgenic botrytis cinerea, transformed with life-actin GFP, and we treated that with 100 micromolar cytokinin, CK. And you can see upon treatment, the life-actin, which is found at the tip in the mock, it loses its tip-specific localization, rather it is diffused throughout the cell. So this could be one reason that the fungus, upon treatment with CK, the pathogen shows an growth inhibition. Also in our transcriptome data, we found that the endocytosis pathway was downregulated. To confirm this, we tried to measure endosome size and density in the hyphae. For this, we used FM464 tracker, which is a very well-known endocytic tracer in the cells, and we found that both endosomal density and endosomal size was downregulated when the pathogen was treated with cytokinin. So this was one part of our research, that you treat the fungus with pathogen with cytokinin, and the fungus is inhibited, the growth is inhibited, its cytoskeleton is disorganized. But that cannot be the only role of cytokinin in the pathogen, because previously people have reported that cytokinins are also produced and secreted by the fungus. So to dig deeper into it, and also if you look at this this phylogenetic analysis, you can see there is a lifestyle-based clustering of CK biosynthesis gene in the fungi. This one for the necrotrophic genes, this is for the hemi or biotrophic genes in the hemibiotrophic or biotrophic fungi. There are some exception, but more or less it's like this. So to dig deeper into it, we did further research, and what we did was we went back to our transcriptomic data that we have generated, and we reanalyzed it. What we found that a lot of energy metabolism pathways were upregulated: glycolysis, oxidative phosphorylation, sucrose metabolism, and it was also reflected in real-time qPCR analysis of some key genes of the metabolic pathway. So what we did, we grew the fungus on the synthetic media, defined synthetic media, varying the concentration of glucose and nutrient contents. And to our surprise, we found that when the glucose availability to the fungus is reduced, the same cytokinin which was inhibitory is no longer inhibitory, rather it was promoting cell growth, fungal biomass. So again, what we did, we treated our life-acting GFP fungus in both rich and 1,4 media. You can see in the graph that the effect of cytokinin is reduced. It's barely significant. It's not that much inhibitory for the disorganization of the F-actin, and the effect is like barely significant. This also shows that when you grow any fungal pathogen in a nutrient limited condition, there is a slow growth. One reason could be this misorganization of life actin, which should be at the tip of the fungus. Since glycolysis was upregulated in our transcriptomic data, what we did, we used our synthetic media with different glucose concentration and nitrogen and phosphate concentration, and treated with 2-deoxy-D-glucose. It's a glucose inhibitor, competitive inhibitor for the glycolysis, and you can see in 1,4-glucose media, CK was able to rescue the glycolysis inhibition in both in second media and in the fourth media. The effect of this in the third media, where nitrogen and phosphorus was reduced, the effect was not seen. We didn't go deeper into it. Maybe it needs better analysis. But when the glucose was reduced to 1/4, you can see CK was able to rescue glycolysis inhibition in the fungus. We also checked the uptake of glucose from the media in different concentrations, and you can see you decrease the glucose concentration, there is more uptake of glucose from the surrounding by the pathogen. How much time I have? 50. Okay. Any effect on the on cell metabolism is also reflected in the redox status. So we tried to see if this effect on the fungal metabolism is also reflected on the fungal redox when we treat the pathogen with the cytokinin. For this purpose, we generated 2 transgenic botrytis lines, one for measuring the mitochondrial redox and one for measuring cytosolic redox. And we treated both redox transgenic line fungus with cytokinin. And we found, this is in vitro result, and we found that after cytokinin treatment, mitochondria was oxidized and cytosol was reduced as compared to mock. So we tried to see whether this effect on redox is also relevant in planta. For this, we used transgenic tomato lines. IPT has higher cytokinin content in leaves, CKX has low cytokinin content in the leaf, and MAT2 was the background. And we treated these different transgenic cytokinin transgenic lines with our transgenic botrytis fungus. And we found that even in planta, in IPT, the mitochondria was more oxidized, and cytosol was reduced. One could say that the less effect on in glucose restricted condition of the cytokinin might be due to less penetration of cytokinin in the fungus. To overrule this, what we did, we treated the fungus pathogen with fluorochrome tagged cytokinin, isopentenyl tagged with NBD, and we found that there is no decrease in the cytokinin penetration both in full media and in the glucose restricted medias. Yeah, okay. So we concluded that there is a dual role of cytokinin plant fungus interaction. And this effect of cytokinin on the pathogen is dependent on sugar availability. Also, after fungal exposure, the plant starts making higher level of cytokinin, which could be inhibitory to the fungus. But at the same time, this pathogen may take advantage of this increased cytokinin to boost its metabolism and prepare itself for the next phase of infection. Okay, by now we know that fungus can sense it is responding to the plant hormone cytokinin. So there must be some agent, some protein helping it to sense the cytokinin. Cytokinin signaling is very well reported in the plants, but in fungi, cytokinin signaling is not well studied. But we know that there is a histidine hybrid histidine kinase that are responsible for environmental sensing in fungi. So what we did, we prepared histidine kinase mutant of the pathogen using site-directed mutagenesis, and we found that the HK mutant, the HK fungal mutant, was not responsive to cytokinin, suggesting that this protein, this histidine kinase, has role in cytokinin sensing in the fungal pathogen. Also, this simply shows that the nutrient changes that were affecting the fungus, the wild type fungus, is not relevant for the HK mutant of the fungus. Also, like we know, when the glucose concentration goes down, the wild type pathogen, when you treat the wild type pathogen with CK, there is a promotion of cell mass, but that effect was not seen in the Botrytis cinerea histidine kinase mutant, further suggesting that histidine kinase has role in cytokinin sensing in the fungi. Okay. This is the effect of this mutation, HK mutation in the pathogen development. There is reduced branching as well as sporulation, and it was also confirmed by qPCR analysis. Also, there was a difference in the cell biology of the mutant. The cell area was reduced, but the cell length was increased. This is like characterizing the mutant, fungal mutant. Also, we did a disease symptom assay, both with the spores and with the plugs. And we found that even in the presence of CK, botrytis, you can see the wild, in case of wild type, CK was inhibiting the infection. But in case of mutant, CK was unable to inhibit the infection of the BCHK mutant. The endogenous CK, we treated the IPT transgenic lines, which has high cytokinin content, and did the infection with the mutant, and we found that even the HK mutant was non-responsive to endogenous plant CK as well. The endocytosis parameter, we now know that the cytokinin affects endocytosis in the fungal pathogen. But since the mutant is non-responsive to CK, so it was the endocytosis parameters in BCHK mutants were not affected significantly. This was just the preliminary work to understand. Just 5 minutes, it will be over. Yeah, okay. Since cytokinin is an adenine, we briefly think that the adenine transporter could also be the transporter for cytokinin in the fungi. For this, we did only just the preliminary results. When we treat cytokinin together with adenine, there is a promotion of cell growth. Both the colony diameter and mycelial drive it. So it could be possible that there is the transporter for adenine and cytokinin is same, but when adenine is present, it is transported in preference to the cytokinin. But this is just a preliminary data, and we are still digging into it. So this is the conclusion. We believe that HK is involved in response to the cytokinin in the fungal pathogen. The HK mutants are less sensitive to nutrient restrictions. It has a role in sporulation and hyphal branching. Also, HK has a role in environment sensing. It was non-responsive to the nutrient stress, and also in botrytis cinerea, light is required for sporulation, and I have shown in previous slide that the HK mutant was not sporulating. So we believe that maybe it's not sensing light. The defective sensing mechanism of the HK mutant might be helping the fungi to evade defense response in the host. And we briefly believe that the adenine transporter is involved in botrytis cinerea CK transport, and HK is downstream to this transporter in the signaling cascade. I'm thankful to my team, Dr. Maya Bar, group leader, my colleague, Dr. Rupali, Dr. Mirav, our PhD student Iftar, our collaborators in Israel, in Czechia, and in Chile. And last but not the least, a big thanks to the funding agencies for financially supporting this project.
So thank you for your talk. Now we have started more than 30-minute delay and wrap up this session well in time. So now the session is open for question. If one or two question we can take up. Okay. Sir, please.
Dr. Gupta, very clean data, clean presentation you had. I was just curious. Is the cytoskeleton in fungi, which you have seen, do they have similarity with, say, mammalian system or, say, human?
There is one group where we are working. They work on plant cytoskeleton. So,
no, what I'm trying to know that, suppose if you look at cytoskeleton, you have tubulin proteins, even actin you have.
Yeah.
So is the amino acid sequences of known fungal cytoskeletal protein are same or not same or dissimilar? Do they behave same way?
The F-actin is different.
No, I mean F-actin is a filamentous actin.
Yeah.
You have the G-actin gets polymerized into F-actin. So that's nothing. The F-actin or G-actin or even the tubulin proteins which you have on those filaments, are they same or different?
In plant and the fungus?
No, plant and, I mean, fungus and mammalian system, say rat and mice, human, whatever.
We think there must be some differences because suppose we are running the qPCR.
Right.
We run a reference gene, standard gene. So if it's different.
No, it's fine. I mean, just curiosity. Yeah, I thought I'll ask you.
I believe it must be different.
And my second quick query was your glucose. When you have used the glucose, it had impact. So are there, say, energy sensors and nutrient sensors? We do have. So in fungal system, is not that glucose sensing is mediated by maybe ADP, ATP ratio and activating the A kinase or that the AMPK or AKT or those kinases, because histidine, you said histidine kinase is a enzyme which can phosphorylate.
Yeah.
So there must be some kinase cascade which might be influencing.
Yeah, downstream the signaling pathway, there is a MAP kinase cascade.
Because there has to be a energy sensor. When you don't have glucose, the ADP, ATP ratio will change. And that will modulate the kinase, which will phosphorylate another one and then give the effect.
It's like there is sensor domain, there is histidine kinase, there is autophosphorylation receiver domain, and then there is a MAP kinase signaling cascade.
Yeah. Thank you very much. So I extend my thanks to all the three speakers. They have very nicely presented. Professor Radha Chaube has presented on the endocrine disruptors, and Dr. Swad has spoken a very strong motivation for students and the teachers also to do bioinformatics and to come up with the good publications. And this shows all these students are now well placed. And this is a very nice presentation. Thank you very much. And Dr. Gupta, you have presented a very nice work. And I wish you early recovery. You got injury here in India? Oh, very sorry. Okay. I wish you will be recovered soon. So with these words, I extend thanks to all the speakers, and I hand over mic.
Thank you very much, sir, for sharing your research on cytokinin response and sensing in fungi. Neeraj, wait. I request our chair and co-chairperson to please facilitate our speaker, Dr. Gautam Anand. Next, I would like to request Dr. Ankita Mishra ma'am to please felicitate our co-chairperson, Dr. Ranjan Mishra sir. Now, I request our chair and co-chairperson to please felicitate our speaker, Dr. Radha Chaubey ma'am. I request chair and co-chairperson to please felicitate Dr. Sinos Scarisus sir. With this, we conclude the presentation for technical session 4. Let's have a big round of applause for our chair and co-chairperson and our esteemed speaker. Before the break, we had an important announcement. After the lunch, we are running 2 parallel sessions, one in Panchakarma and other one